Hey everybody, we are going to start chapter 5 and we're going to be looking at the structure and function of large biological molecules. We call these macromolecules, macro meaning large, and these are large molecules that are formed by joining many subunits together. So we also call them polymers because again poly means many, so it's many subunits linked together. And you link together monomers, which those are the building blocks of a polymer. So if you were to look at this as a polymer, this brick wall, then the monomers would be the individual bricks themselves. So we make polymers through something called dehydration synthesis. This is the chemical reaction that joins monomers into polymers. You have covalent bonds that are formed by the removal of a water molecule between the monomers. And then the opposite of that, or the breaking down of polymers into monomers, is hydrolysis. And so hydro from water, and then lysis from lyse to split. It's using water to split or break polymers into their monomers by adding water. So this is a diagram of both. So here you have a short polymer and you want to add this single monomer to it. You can see you have an OH group or hydroxyl group right here and then a hydrogen. So together those make H2O, which is water. So the water will leave and now you have a fourth monomer linked together. And then to break that apart, you would simply add water back in. You recreate the hydroxyl group over here, the hydrogen over here, and so you have broken that polymer into, again, the three and then the single monomer. So when we get into some of these large macromolecules, we're going to focus on four classes. We're going to talk about carbohydrates, lipids, proteins, and nucleic acids. Today we're going to be focusing on carbohydrates. So for each of the classes of macromolecules, make sure you guys pay attention to the elements it contains, what the monomer units and structures look like. You should know some examples, and then you should also know what their use or their role is in nature. So carbohydrates are used for fuel, for building materials, and as receptors on cell membranes. So carbohydrates, if you look at the name, you've got carbo, so it's made of carbon, and then hydrate sounds like water, and so it's made of hydrogen and oxygen. And it's actually interesting when you look at the general formula, it's always going to be CH2O, meaning that you're always going to have the same numbers of carbon and oxygen, and then twice the number of hydrogen. So we're going to talk about four different types of carbohydrates. We'll start with the simplest, which are the monosaccharides, then disaccharides, oligosaccharides, and finally polysaccharides. So monosaccharides, mono meaning one, saccharide meaning sugar. So these are going to be very simple sugars and they'll contain about 3 to 7 carbons. And these can actually be linear, so it can be a carbon chain, or they can be in ring form. So like it'll actually look like a ring when you see it. And these can be aldoses or ketoses. So we talked about aldehydes and ketones this past week. And so it all depends on the location of that carbonyl functional group. So here we have the aldoses. So you can see that carbonyl group is on the end of a carbon chain. And then with the ketoses, the carbonyl group is in the middle of the carbon chain. So now some examples are going to be glucose, galactose, ribose, and fructose. And hopefully you caught on that they all ended in OSE. So when you see that ending, you're pretty much going to know that you're looking at a carbohydrate. Next, we've got disaccharides, di meaning two, and these are sugars formed by joining two monosaccharides through a certain bond called a glycosidic linkage. So these glycosidic linkages are going to form, so you can see right here, you have a bond formed between the first carbon and the fourth carbon of these two, they were over here, two glucose molecules, so two monosaccharides. And now we've formed a disaccharide called maltose, and it is a 1,4 glycosidic linkage because it happens between the first carbon and the fourth carbon, okay? And then down here, you have glucose and fructose, and they will join together to form sucrose, and here you have a 1,2 glycosidic linkage. Some more examples, maltose is two glucose together, lactose is a glucose and a galactose, and then a sucrose molecule is when you add glucose and fructose, or join glucose and fructose. All right, next we've got oligosaccharides, and this is going to be when you join 2 to 10 simple sugars together. So larger than disaccharides, but not so huge that we would consider them polysaccharides. And these are used in cell membranes. And we'll talk about those a lot more actually in the next unit. All right, so then polysaccharides, this is going to be many simple sugars joined together, and these are going to be used for storage or for structure. So a few examples are starch, cellulose, and glycogen. Here's a diagram, or two diagrams, I guess. You've got starch up here in two different forms, and then you've got glycogen down here. Something to keep in mind is there are actually two forms of glucose, alpha glucose and beta glucose, and so you can see it has to do with where this hydroxyl group is positioned. So when you are looking at starch, you have 1 to 4 linkages. So it's a 1 to 4 glycosidic linkage of alpha glucose monomers, and then with cellulose, it's a 1 to 4 linkage of beta glucose monomers. So there is a difference there. So starch is going to be made of those 1 to 4 glycosidic linkages of alpha glucose. And the way that this linkage works is it makes the molecule form a helix. And then this is going to be used as fuel storage in plants. So this is how they store sugars that they need for energy. Cellulose then is made of 1 to 4 glycosidic linkages of beta glucose. And this linkage, this type of linkage, makes it a straight line. And this is going to be used for structure in plant cell walls. What's kind of interesting is that most organisms can digest starch, but very few can actually digest cellulose. So this is a pretty important example of the link between structure and function. And then our last example is glycogen, and this is essentially like animal starch. So we say that because it's similar to starch, but it has more 1 to 6 linkages or branches. And this is commonly found in liver and muscle cells. All right, so your lecture question for today. Why is it beneficial for plants to have cellulose in their cell walls? So think about the differences between cellulose and starch and some of the little tidbits you got about the two molecules, and try to think about an answer. Why is it beneficial for plants to have cellulose in their cell walls? Maybe instead of like what would be different if they had starch in their cell walls? So maybe compare it that way. And please don't forget to send me an answer to your lecture question via the website. Thanks, guys.